Module 5, we look at how to understand the input and output files from MateCell, so we can get an understanding of how MateCell is actually choosing the progeny, whether the progeny and matings that MateCell has chosen actually align with our breeding objective and whether they're appropriate or whether we need to add additional parameters on top to optimize the joining and mating decisions that MateCell's made. So in terms of the input files, this is really important to get correct before you actually start your MateCell runs. So very important that you need either a text file or a CSV file. These files need to be split by sex, so you need to have a RAM list and a U list separately in each text file or CSV file, and then also have individual files by joining groups as well. So if you have U's that you want to AI, but also naturally mate, you need to create two different files for those U's. You also need to make sure that every animal in the file has the 16-digit sheep genetics ID and that those animals actually exist in the sheep genetics database. MateCell leverages and talks to the sheep genetics database to look at their optimal joining decisions. So you need to make sure that those animals exist, otherwise MateCell's not going to work for those particular animals. And in this step, you can also upload either a custom index or any research breeding value that you have, or pole horn, for example. So in the text file that you create, you have the first column, which is the animal ID, and then the second column, which is potentially the custom index or the research breeding value as well. So in terms of the joining groups and parameters, we will run through this and where this is all found on MateCell when we do a practical demonstration. But there are a few things that you need to think about in terms of the inputs before actually starting the MateCell run. So the grouping section is essentially the practical logistics of what's actually going to happen on farm. As I mentioned, if you're, say, doing an AI program and a natural mating, we need to make sure that the groupings are correct so that MateCell is looking at the best potential joining outcomes based on the logistics that are actually happening on farm for that particular mating period. So there are a few other options that you can specify under the grouping section, and again we'll run through this during the demonstration, but the min use section is the minimum number of U's a RAM can receive if that U is selected, and likewise for the minimum number of RAMs that a U can receive if selected as well. The max use is the maximum number of U's that a RAM can receive if the U is selected. And then the absolute minimum use is the number of, the minimum each RAM must be actually used. So this is really important to think about if you have AI or ET programs, or you're really chasing an absolute minimum number of animals from that particular RAM during a natural mating as well. So a little bit different, and MateCell gives you those definitions during the run, so you don't have to necessarily remember them off the top of your head, but it is really important to know the difference between them. Another thing that you can also overlay before actually starting your run is parameters, and this is where you put your thresholds for inbreeding, as well as, I guess, your target kind of rate of genetic gain as well. So that strategy around genetic diversity and also genetic gain. And we'll go through that during the run and how to input those. So in terms of the outputs that MateCell provides you, there are a few different output sections that MateCell gives. So this is looking at the frontier. So essentially the graph that MateCell provides you after it's finished running. And this frontier looks at the potential genetic gain, obviously using an index, as well as the level of co-ancestry of the expected matings that MateCell has looked at. So there are a few things to think about in terms of this frontier. We have genetic gain on the y-axis and then co-ancestry on the x-axis. Where we get further away or further onto the right-hand side from the x-axis, that is less diverse. So a higher co-ancestry, where if it's closer to if it's closer to the y-axis, then that is higher co-ancestry, so higher diversity there. The blue line that's on the front here is essentially the frontier line. So that's the level of co-ancestry at the level of genetic gain. So for each one of those dots, that represents the level of co-ancestry where it coincides with the genetic gain that is potentially an option for the mating. The green line tells you the pressure that you've actually placed on co-ancestry. So for each one of those dots on the blue line, if you have, for example, placed 10 target degrees on co-ancestry, the green line will intersect with the first blue line on the front here. It's a little confusing, but we can go through it during the run as well. So the orange dot is essentially the end result. So if you've specified 10 target degrees, that green line will hit the first dot of the blue line, and then your end result will be around that point. And then the red line on the front here is essentially the path that MateCell took. So MateCell runs lots of iterations to come up with the best potential outcome. So it's just showing you roughly the path that MateCell took. So the main thing to kind of think about here is the blue line, the green line, and the orange end result as well. So if we talk through this with a bit of an example, this example looks at high rate of genetic gain, but also high amount of genetic diversity in the progeny outcome. So we have a nice frontier graph that's managed to make a high rate of genetic gain, but also potential to place more or less emphasis on co-ancestry. So this particular example would have been run with animals that were lowly related, more likely lowly related candidate males with a similar index. So most of the males in this run would have had a high index and would have been lowly related. So for this particular run, we actually didn't have to sacrifice much genetic gain based on the level of co-ancestry, because there was you didn't have to go back. If you wanted to place more emphasis on co-ancestry or making the progeny more genetically diverse, you did not have to sacrifice a lot of genetic gain. So if you wanted to place less emphasis on co-ancestry, you could make marginal rate of genetic gain, but this is a great outcome essentially for MateCell, because you don't have to actually sacrifice genetic gain, but can push quite a lot of emphasis on ensuring a lower amount of co-ancestry in the expected mating. So for this particular example of the output for MateCell, this example looks at a lower rate of genetic gain in the expected outcome, but also a higher rate of genetic diversity. So in this example, there probably would have been fewer male candidates with a high index, and those male candidates would have been quite highly related. So to essentially ensure that we're creating more diversity in the population, we actually had to sacrifice quite a bit of index to get to this point. So to reduce co-ancestry, the index also had to be reduced as well. In terms of other outputs, the console tells you, so on the right-hand side of the screen, all of that text in the white box, that's the console output. That actually provides information as MateCell's running to tell you essentially how MateCell has run. So if MateCell's run successfully, we will get a job done line, and that's right at the bottom of the console output. If MateCell hasn't run successfully, then it will tell you in this console output why. So that's really important if you are coming stuck running MateCell, trying to figure out exactly why, whether that's something to do with your input file, whether you've accidentally put too few or too many backup RAMs, for example. There's a whole heap of reasons why MateCell might not run. So this console output's really important to check to make sure that it's actually done the job successfully. And if it hasn't, why hasn't it? And if you don't know, you can always contact Cheap Genetics to figure out the error message in the console, essentially. Sorry, the other thing that you probably should double check in terms of the console output is this parameters section on the left-hand side. So you want to cross-check the number of matings to be generated is actually what you inputted in the setup of MateCell. and also the target degrees as well, that's actually aligns with what you set Matescell up to do. In terms of the candidate section of the output file, you want to go through and just double check that it's all worked correctly based on the setup that you've defined in Matescell. So you want to check the candidate section to make sure that the male candidates was actually correct based on the number of RAMs that you inputted. The female candidates was correct, and that the selected both size and DAMs aligns with the requirements and definitions that you put into Matescell. So if you defined that Matescell, for this example, needed to create 500 matings, something would have happened here because the selected DAMs and the matings used was only 330. So you need to then go back and find out what happened and why Matescell only decided to choose 330 RAMs, not DAMs, sorry, not 500. The other section to have a look at is the index constraints. So have a look at the average index of the progeny here. Is that high? Is that lower than what you're expecting? Look at the lowest index selected, the highest index selected, just to see what the range is, and then also the standard deviation of the progeny as well, just to see how much you can either tighten that or increase it if you need to. We can also have a look at the targets. So it's always really good to review the fitness. So the fitness is essentially a term to specify how well Matescell achieved what you specified in the run inputs. So obviously the higher the fitness, the better the result. Also have a look at the target degrees against the achieved degrees. So if you defined that you wanted to put 10 target degrees on co-ancestry, if the achieved degrees was higher or lower significantly than what you specified, then it would be worthwhile going back and checking why that's the case and rerunning again. And that's similar to for the target and achieved co-ancestry as well. So just making sure that those two numbers of the target and achieved for both the degrees and co-ancestry are similar. And if not, either contact sheet genetics or have a look through what you defined in the input and the parameter setup to make sure that they're as close to possible. The other thing that will be available on the output section of the console is the inbreeding constraints. So wherever you see F in Matescell, F refers to inbreeding. And it's really important to review the progeny inbreeding. We looked at the potential co-ancestry in the last slide, but it's also important to have a look at the progeny inbreeding as well. So in this example, there was a weighting placed of -1 on progeny average inbreeding, and then we can have a look and see how that was achieved, and also the maximum progeny inbreeding achieved as well. So really important to review that to see if you need to place some larger constraints on inbreeding or co-ancestry in your next Matescell run. Matescell will give you an Excel file to download, so this will be available on Matescell itself, but also in the out matings files that are emailed to you automatically after you successfully run a Matescell result. So it's a bit hard to see on the screen here, and we can go through it in the demonstration, but essentially this is the mating list that Matescell provides. So it's the sire and dam that Matescell has chosen based on the inputs that you've specified for the best optimal mating that you've specified. So it also then has a look at the mid-parent breeding value for the sire and dam for a whole heap of traits. So you can go through, check how many sires have been joined to dams and whether that's like appropriate based on your breeding objective, but also based on logistics of the on-farm matings. So really important to, I guess, review this. And there's a few easy ways that you can do this. We've developed some easy tools so you don't have to scan through and overlay calculations and graphs onto this Excel if you don't want to. So one easy way to have a look at the distribution of progeny that Matescell, or the matings that Matescell has, I guess, specified, is to load that Excel that you get from Matescell into our Matescell results tool app. So all you'll need to do is essentially either drag and drop or click browse on the Matescell tool app and find the file that Matescell's spat out for you from your out matings. And you can also have a look at, I guess, a few distributions and graphs here to make sure that mate cell's actually doing what you want it to. So on the left-hand side of the screen on the Mate Cell Results Tools app, there'll be a few different graphs and summaries that you can actually have a look through to make sure that Mate Cell's done its job correctly. So one of those graphs is the progeny distribution graph. On the top left-hand side of the screen, you can select the trait, and this graph will essentially plot the distribution of progeny or the expected mating outcomes against the density for that particular trait. So for example, if increasing birth weight was important to your breeding objective, you could have a look at where Mate Cell identified the most optimal progeny outputs for that particular run. And if you wanted to increase the mean slightly, you could go back into Mate Cell and then place a little bit more emphasis on increasing the mean for birth weight. So that's, I guess, when we were saying before that you need to run Mate Cell, do a basic run first on the index, see what the outcomes are for that, and then you can go through and refine the individual traits from there. Another graph that you can have a look at through this Mate Cell Results tool is the outputs of the sire usage. So it will tell you the sires that Mate Cell essentially identified that were the best to result in best and most optimal progeny outcomes, and also the number of times that sire was used. So it's really important to cross-check this result here against what you've actually defined in your Mate Cell setup to make sure that Mate Cell isn't using less or more than what you defined. It's really important too to go through and have a look at some of the output files that are included in that email that Mate Cell sends you after the run. So if you wanted to have a look at any backups, and if you specified backups in your Mate Cell setup, you would go to the backup file, which is a text file that you can open. And this has a whole heap of information, including the dams, the primary sire, and the backup sire as well. So if we have a look at what this backup file is defining, so we've got the dam ID, we have the number of backups which was defined in the input settings in Mate Cell that you would have defined before your run. We have Mate Cell's first RAM choice, or the primary RAM ID. We have the F, so F, remember, is always inbreeding. We have the progeny inbreeding of Mate Cell's first RAM choice based on that dam that Mate Cell's also linked with that RAM. And then we have the backup, so Mate Cell's secondary choice, essentially. And then we have the progeny inbreeding of Mate Cell's secondary choice.